keynote, I would just like to say a few more words about how CAMDA works, because it's a little bit different to most of the sessions that you will see at ISMB. And I'd like to briefly introduce the 3 contest challenges that form the backbone of CAMDA. So every year we advertise between 2 and 3 data analysis challenges that then scientists worldwide take up and present their results here at the conference. So for 2017, the challenges included a Neuroblastoma data integration challenge by the MetaSUB consortium, by Matthias Fischer's lab, the MetaSUB Inter-City challenge by the MetaSUB consortium, and the Oxford Nanopore Wiggle Space challenge by the lab of Chris Mason. And we sort the presentations according to these different areas. And we will end the conference with an award ceremony where we will present the prizes that you vote for. You vote for the best presentation in this open-ended data analysis contest. And we will discuss future contests. So if you have particular areas or questions that you think are really cutting edge and are in need of multiple people looking at them at the same time, please take part in the panel discussion that we will have on Sunday afternoon. I'm going to say a little bit about each of these challenges. So today, the presentations will start off on results people had on the Neuroblastoma data integration challenge. This is data coming primarily from the lab of Matthias Fischer from Cologne. Neuroblastoma is the most prevalent extracranial solid neurological tumor. It develops from immature nerve cells, actually most often around adrenal glands, which have cells that are similar to immature nerve tissue, which sits atop the kidneys. And it mostly affects children that are young, 5 years or younger. And the striking situation for these cancers is that the prognosis varies highly. So in some children, the cancer more or less disappears by itself after a while, and in others, it is a highly deadly disease. Symptoms include lumps. For instance, at the original location of the primary tumor, pain, bruising around the eyes, and the challenge included both clinical data that was made available and gene expression data from 2 complementary platforms, both RNA-seq and from Agilent microarrays. And this year, we actually got additional data that was matched to the gene expression in the clinical data by a master table, informing us on copy number variation. So we're basically looking at a subset of the hierarchy of different data types that Ludwig Wessels introduced as part of this challenge. And obviously, the question was, can we improve the prediction of clinical outcome? Can we perhaps say something about survival time or find something about disease mechanisms? And again, I stress that the way CAMDA operates is not by having a known goal and then trying to see whether you can improve prediction performance from 86 to 87% or something like that. But we want to encourage and reward people who take unusual approaches and see things in the data that other people haven't seen. So this is the contest that we will spend most of the time today discussing. Then this session will be followed by a number of flash talks about cutting-edge work that we will go on to discuss in great detail on the poster session, because you will see that with the many contributions, the time for discussion right after the talk is limited. So for things that are very, very new and lend themselves to further discussion, we will have a very intense session at the end of the day where we'll discuss both results from the neuroblastoma and cutting-edge work on the second challenge, which is the MetaSUB Inter-City challenge. This is coordinated and collected by the MetaSUB consortium, where people basically walk around different cities internationally and take little swabs and take DNA samples across different locations, and particularly in the subway, in public places, in the metro, so say from a ticket machine or from a train carriage, and then analyze them for the organisms that they find there. So we get a cross-section of urban microbiomes from 3 different cities in this particular challenge, collected from different surface materials like wood or metal, and a range of public locations. And the challenge questions include questions like, to what degree can we identify and rediscover organism fingerprints? What can we say about the variation of biodiversity in these samples? And what type of insights could you derive from functional analyses? Last but not least, we included a challenge that we call the Wiggle Space Challenge, where we basically try to understand better and exploit better data that comes from Oxford Nanopore technologies. That's a next generation sequencing technology, where basically a long nucleotide is pulled through a nanopore, affecting the capacity and the current that you can measure across this pore. And as the nucleotide is pulled through, that gives rise to these characteristic wiggles. And we actually provide the raw data of these wiggles to basically let people find new ways of dealing with this. Actually, a couple of years ago, someone in Switzerland was not trying to convert the wiggle data to sequence space, but actually did it the other way around, converted sequence databases to wiggle space, and then was comparing the sequenced wiggles to the database wiggles by music search programs, and that worked surprisingly well. So we're trying to encourage, again, different new type of thinking on the data. So we have these raw data from one of the latest next generation sequencing platforms. We have crucially matched sequencing results from more established platforms like the Illumina. And to add to the mystery, we have a sample of patient microbiota that we haven't disclosed yet what it is. The question, of course, is, can you find out? Can you find new ways of computation in wiggle space? Can you calibrate your methods using these matched data? And of course, what can you find out about these long read data sets that we provide? At the end of this CAMDA conference, we will discuss interesting ways forward, possible new data sets, possible new questions from a strategic point of view, basically trying to think ahead what the future of our field might look like. I would say it will not look like this, but I needed some visual aids. So we will have a striking keynote introducing the question of reproducibility in our field and linking that to the blockchain by Cesare Fornello. Then we will discuss possibilities of looking at forensics using metagenomic methods, human exposome, drug liver toxicity, where we collaborate with the FDA in the US, and different strategies in precision medicine. We will invite you to contribute ideas and suggestions regarding the community challenges for the next year and beyond, including a discussion of the introduction or limitations of non-human model systems for precision medicine, and difficult disease contexts like chronic fatigue syndrome. So I hope you will enjoy this very varied conference track at ISMB as much as we do, and wish you a very lively session. So we start with the first speaker, please.